Hi, my name is Alex, and today we're going to be talking about lateral root growth. Lateral roots begin to grow after about 5 to 10 days after germination occurs, and they grow after the primary stem reaches about 2 to 3 centimeters in response to different water and nutrients.
There are 4 main phases of lateral root development. These are 1. Positioning, 2. Initiation, 3. Outgrowth, and 4. Emergence. Lateral roots develop from specially marked xylem pole pericycle cells. The 12 xylem pole cells are arranged in a circular layer around the xylem. The main root is divided into 4 different zones: the meristem, the transition zone, the elongation zone, and the differentiation zone. Lateral root growth won't occur in the phloem because inhibitory enzyme IC2 inhibits lateral root growth when auxin is not present, much like the ethylene receptors inhibit the triple response when ethylene is not present. And now we'll be talking about lateral root formation and the first step, positioning. Auxin plays key roles in the lateral root growth, and the accumulation of it is necessary to begin this process. Auxin is produced from the tryptophan pathway and is produced from precursors like indole-3 butyric acid, IBA, and indole-3 acetic acid, IAA. Naxalin facilitates the conversion of IBA to IAA in root caps. This occurs in the outermost cells of the main root cap that are dying. Auxin created in the root cap cells is funneled inward to the oscillation zone. The oscillation zone includes both the transition zone and the elongation zone. Simultaneously, a promoter called direct response 5, DR5, is expressed at different levels in each of the pole cells of the OZ. The DR5 promoter is fused to the Gus reporter gene and is observed in protoxylem cells. DR5 indicates which xylem pericycle pole cells will ultimately become lateral root founder cells. Pole cells with high DR5 expression are selected for this special function.
To provide a visual aid for the oscillations of DR5, we have Jordan Dewey jumping jacks at cells with high DR5 expression and motionless at cells with lower DR5 expression. She's starting in a cell with low DR5 expression and moving to the cell with high DR5 expression.
After discussing positioning, we are now going to be moving on to initiation. Now that we've determined which cells will become our lateral root founder cells, they can begin undergoing mitosis. Cell nuclei begin moving more centrally during this time, and about 4 to 7 hours after relocating, founder cells begin dividing. The enzyme cyclin is a key contributor to initiate this transition. Cyclin helps cell transition from the G2 phase to meiotic phase. There are multiple pathways to activate lateral root initiation. We will talk about 2 of them. The first pathway acts in a similar way to the triple response pathway. When auxin is present, it deactivates solitary root proteins. Solitary root proteins are negative regulators of the lateral root initiation pathway and work to inhibit the auxin response factors, ARF, transcription factors. When the solitary root proteins are inactivated, ARF7 and ARF19 are free to switch on lateral organ boundary domain proteins, or LBDs. LBDs latch onto the promoter of E2FA gene, which permits the first asymmetric divisions to occur. Shoot hypocaudal 2-protein Shy2 is a key player in initiating division. Its presence inhibits the growth of cells in the lateral root. When it is degraded, division is promoted in founder cells in order to compensate for the empty space left by the loss of Shy2. This causes the founder cells of the lateral root to become bigger. In addition to playing a role in the positioning of lateral root founder cells, auxin also facilitates founder cell growth. PIN1 transports auxin to the lateral roots in a similar way that it transports auxin in the shoot apical meristem. PIN aligns between endodermal and founder cells to facilitate the transport of auxin from the endodermal cells into the founder cells, which facilitates founder cell mitosis. Similar to spacing of leaf primordia and the shoot apical meristem, the primary root also has mechanisms to ensure that the lateral roots are spaced far enough away for maximal nutrient and water absorption. As the primary root grows, it curves in response to different nutrients and water availability. These curves are in convex and concave shapes, causing the root to stretch in certain places. The convex side of xylem pole pericycle is more stretched and tends to collect auxin and become an auxin sink because of this. To provide another visual aid, Alex and Jordan are out running on the track. Just as the runner on the outside lane covers more distance than the runner in the innermost lane, the convex side of the xylem pole pericycle stretches more than the concave side, and consequently, the convex side catches more auxin. The ALF protein sequence is involved in monitoring lateral root differentiation through its varying positive and negative regulation. The first protein in the pathway, ALF1, monitors the level of free IAA in the lateral root and regulates the number of lateral roots that will form by sequestering free IAA. ALF4 is inhibited by the ALF1 protein, which is an IAA-sensitive cell within the pericycle. After the lateral root grows to a certain size, it begins to become insensitive to the IAA produced in primary root and must produce its own. ALF3 activity helps facilitate this and is promoted by ALF1. Now that we've covered initiation, we can move on to the outgrowth phase. Initially, lateral root founder cells seem to divide periclinally. As mentioned in lecture, periclinal divisions produce new cell layers. The periclinal divisions of these new lateral root founder cells generate the same layers that we see in the primary root meristem, such as the QC. After 3 to 5 cell layers are created, the lateral root primordia becomes independent. The lateral root first forms parallel to the main root axis before swinging out perpendicularly. As always, the plant has strategies to prevent the waste of resources if circumstances are not ideal for lateral root growth. For example, cytokinin controls the flow of auxin by controlling the presence of PIN1. If conditions surrounding the plant are damaging to DNA, suppressor of gamma response 1 and cytokinin team up to inhibit the outgrowth of lateral roots. After finishing the outgrowth phase, we finally got into the emergence phase. Lateral roots have one final barrier to cross before they are free to collect nutrients. They must burst through and protrude from the layers above them. There are 2 different fates for these overlying cell layers as lateral roots liberate themselves. The cells are either flattened and killed or spread apart from their neighbors. The goal of both of these methods is to generate a hole for the primordia to pass through. The endodermal cells are flattened as the primordia pushes out until they are compressed so much that they die. On the other hand, the cortical and epidermal cells are disconnected and pushed to the sides. To help push these layers apart, HASA and HASA-like 2 kinases degrade the pectin that holds the cell walls together. Additionally, water flow to the primordium increases because water is prevented from traveling into cortical and epidermal cells. Again, auxin is the star of the pathway as its presence negatively regulates water transport through aquaporins. Thus, the cortical and epidermal cells shrink in size, much like water balloons losing water, and are separated from each other as pectins are degraded. This creates a large enough passageway for the newly formed lateral root to emerge